Hello and welcome to Progressive Education Society's Modern College of Pharmacy, Nigri, Pune. In the subject Pharmacognosy and Phytopharmaceuticals for SY Pharm. Myself, Dr. Bhushan Pimpre. In this video, we will discuss about Nux vomica seeds, the morphology as well as microscopical aspects of these seeds. About the Nux vomica seeds, these are also called as poison nut or semen stretchness or Quaker buttons, or in Marathi is well known as kuchala. These seeds are obtained from a plant which grows up to a height of 10 to 15 meters, and the biological source is dried ripe seeds. Ripe seeds in sense the seeds collected from ripe fruits of Strychnos nux vomica Linn. Linn is the author, belonging to family Loganiaceae. The major chemical constituents of this plant are strychnine, brucine, alpha-colubrin, beta-colubrin. These all are alkaloids. They end with a suffix -ine, whereas it also has one glycoside, that is loganin. That generally the glycosides end with the suffix -in. It has various medicinal properties, but initially in the British era, it was used as dog poison to kill the stray dogs. It is also used as a pesticide to kill various rodents of the warehouses, and in laboratory to induce catalepsy in the laboratory animals. Catalepsy means seizures, or in simple sense, fits. If you want to study the effect of anti-epileptic drugs, that means the drugs which are to be used for the treatment of fits or seizures. In that case, ज्याला आपण मराठीत दात खिळी बसणे म्हणतो, that kind of diseases, if you want to treat those kind of diseases with the drugs you have isolated or prepared in the lab, first you need to produce catalepsy in the rats. Rats and mice very rarely they suffer from this disease, but the moment you inject strychnine chemical into the rats or mice, they suffer from this disease. This is called as inducing agent. It induces the disease or disorder or a condition in the rat. That means okay. And then you can try your isolated or synthesized drug. In homeopathy, these seeds they find very important role, and the low doses of these seeds are used, although it is a poison, but in very low doses it has been found to treat anger, irritability, or even constipation and vomiting. So these drugs are in generally homeopathy, they follow very low dilute or dilutions or very dilute solutions of the drug. Even in Ayurveda, the kuchala is found to be used in various formulation, but at a very low doses. About the morphology of the plant seed, the seed appears to be disc shape. Disc shape means it is round but flat. If you observe it from the sides, it appears to be very flat, like that of a 5 rupee coin. Whereas from this is the bottom view and this is the top view. Now these the color of the seed is grayish green, and odor, it does not have any odor. Taste is extremely bitter, but I will suggest not to taste this drug because it is not a direct poison, but it will definitely cause vomiting or diarrhea, or even um, problems related to GI tract. Severe vomiting can happen due to this. So these drugs or the seed should not be tasted. But it is extremely bitter because of the presence of strychnine and brucine, which are alkaloids. Size of the seed, the diameter is around 2 to 3 centimeter, and the thickness is around 0.5 to 1 centimeter. Shape is ovate or round. Special features. These seeds, they have very fine hair on the surface, and these hair are called as trichomes, and they provide a velvety appearance to the seed. So if you hold the seed between the two fingers and move your fingers, you will notice it is too smooth, like that of a fur of a very small animal, and it has a prominent ridge at the periphery. You can touch this drug. It is not toxic if you touch it, but it is not recommended to be consumed. Now about the transverse section. As you can see here, the seed has to be held in horizontal direction so that the ridge faces towards ourselves, and it has to be cut in this way so as to you get this kind of sections. Now, these two cotyledons, they fuse with each other to form a ridge. Ridge means a bulge, a very fine bulge over the at the place of joining. About the microscopy of this seed, once you take the section, if you observe under microscope, it shows various characteristics or various layers. The first layers, these layers are grouped into two category. Since it is a seed, it is the outer category or outer layer is combining together. It is called as testa, whereas the inner layers are called as endosperm. Testa is the uppermost layer, which has three sub layers into it. Now we'll see one by one the trichome. The trichome are hair-like, smooth and velvety structure, and these are lignified in nature. That means they will be stained pink on treatment with fluoroglycinol HCl. So these are lignified trichome. Just below the lignified trichome, you can find the single layer of epidermal cell. Most of the epidermal cells, they are modified into trichome, whereas few which are not converted into trichome, they remain as flat, tangentially elongated. Now about the parenchyma. There is a presence of parenchyma below the epidermis, but it has been collapsed. Collapsed in sense it has been constricted or pressed from both the sides. The inner as well as outer side presses the parenchyma, and that is the reason why it forms a very thin layer, single layer. You can differentiate between these layers only after staining with fluoroglycinol HCl, because the epidermal cells and the trichomes, they show pink color, whereas the collapsed parenchyma, it shows yellow color. We will have a look at it in the later slides. Next, about the endosperm, the lower region is called as an endosperm. Now this endosperm of any of the seed is the storehouse or storage of food material for the young one. Now the seed is very analogous to an egg. Like in egg, uh, there is a presence of protein. Now what does that protein is meant for? This protein helps the young one eats up all the protein or develops from this protein and breaks the eggshell and comes out. Very similarly, very similarly in seeds also, you will notice the presence of protein, oil. Now we will see the oil globules. These are oil globules. They are minimum 1 or 2 oil globules per cell. Then the proteins which we were talking about are these. Every cell has abundant amount of protein into it. This protein is taken up by the endosperm, embryo. Embryo is residing after this is I have drawn the structure only of a single cotyledon. Now here will be the gap or the embryo will be present, and just below that again you will find the mirror image of the upper cotyledon. So this is the small part of upper cotyledon. Now plasmodesma. Plasmodesma are connecting channels. These are very similar to tunnels that join the two cells. Why they are joining the two cells is because the embryo is present here and it can derive nourishment from the adjoining cells. But what if or the adjoining cells are exhausted of the protein or the nourishment? Then in that case, the upper protein flows from one cell to another or towards the center through these plasmodesma. Plasmodesma is the plural, plasmodesmata is the singular. These are made up of cellulose or the material similar to the cell wall. Besides, the plant has definite location for the alkaloids, especially brucine and strychnine. If you have a look at this brucine, it is located at the peripheral cells. Peripheral cells, they show the presence of brucine alkaloid, whereas the central cells, they show major amount of strychnine in them. These can be identified only after staining the tears. So this is the rough idea about the microscopy of the Nux vomica seed. Then regarding the staining of Nux vomica seeds, you have to use minimum 4 to 5 different stains to determine different cells in the tears. Now, since we have discussed about the trichomes are lignified, and upon staining with the difluoroglucinol HCl, it stains pink color. It gets pink color. So, fluoroglucinol HCl gives pink color to the lignified trichome. So, this is our first dye. Second, Sudan red. As we all know, Sudan red is soluble in oil. It is oil soluble dye. So it will specifically stain only the oil globules. Next, about the picric acid. Picric acid, it forms a bond with proteins and it gives yellow color to the protein. So the proteins here are alluron grains. Alluron grains are proteinous in nature. So if you stain the TS with picric acid and wash it out, you will notice the change in the color. That means the alluron grain, they get stained yellow in color. Next, about the plasmodesmata, as we have seen, these are cellulosic, hemicellulosic strands, and cellulose is nothing but carbohydrate. So if you break down the carbohydrate with H2SO4 hydrolytic reaction and then stain that with iodine, these are made up of starch granules, so they take bluish color. But only after treatment with H2SO4, you should treat it with iodine. So this is how the plasmodesmata appears. And if you wash it many times, there are chances that the iodine will get washed out. Then about the strychnine and brucine. Remember, brucine, it gets stained orange in color on staining with or on reacting with HNO3. Now, before you will notice here the cells which I have shown has only brucine, and I have not shown the protein as well as oil globule in these cells. This indicates we should first take out the protein or we should first take out the oil globules from these cells. Now, how to take out the oil globules is take the section and uh, shake it with petroleum ether or N-hexane. Petroleum ether or N-hexane, they take out the oil globule. Once the oil globule goes out of the cell, then and then only this aqueous acid will be able to enter into the cell. So if oils are present, acid will not enter. So the first step in conducting the reaction of brucine is to shake the thick section of Naxomica with petroleum ether and then keep it in concentrated or fuming HNO3. It shows orange color. Whereas if you stain similar section, that is the section which is devoid of oil with ammonium vanadate, it will give purple color. Now I have shown all the colors with very high intensity, and the reason for this is most of you are using different kind of screens. Some of you might be using laptops or cell phones, so every individual will not have all the colors, all the shades of the color. So I have chosen the brightest color which is able to be visible everywhere. So most of you might feel that the upper one was not an orange color, but it is true. The color is brighter, purposely chosen by brighter. Okay, so these are the color bars. Once we will again revise them, fluoroglycinol will give pink color to the trichome. Sudan red will give reddish or color to the oil globules. Picric acid will give, or it will go and stick to the protein. So alluron grains will be yellow in color. Iodine will stain the stars present in the hemicellulose of plasmodesmata. HNO3 will stain orange color to the brucine, and ammonium vanadate will give purple color to the strychnine, which is located at the center. Now we'll have a look at the actual. Now this is about the testa of the plant, and here you will notice that the trichomes are lignified in nature. Uh, they are unicellular and they have a bulbous base, a bulge or a bulb-like structure is present at the bottom. So all the trichomes are moving in a common direction, and just below the trichome you will notice the presence of a band. This band is of epidermal cells which are also lignified, and few of these epidermal cells have been modified into trichome and few are still present, so they are uh, can be differentiated from the trichome. Just below that are the collapsed parenchyma. Now you can notice here the columnar cells which are shrunken and are yellowish color, a yellow colored band or golden band below the epidermal cell, that is called as collapsed parenchyma. Now upon staining with picric acid, the alluron grains they take up the yellow color, and this is the slide that is observed under for 10x objective and 10x eyepiece. So you can notice the cells, so few of the alluron grains they are migrating from one cell to another cell. Whereas the slide on the right side is taken at 40x objective and 10x eyepiece, wherein you can notice the shape of the alluron grains, which is boat shape or tapered from both the sides. Now, these are the images of oil globules attached with the alluron grains. So they are present very closer to the alluron grain. The oil globules can be stained pinkish in color with Sudan red 3. Now the background is also pinkish in color, so you are unable to notice the difference between the color of oil globules. But in the microscope or with the naked eye, you will be able to observe clear or distinct pinkish color. These are the plasmodesma. Plasmodesma are, as we have seen, they are connecting strands, and you can notice here that the there is a tunnel between the two cells which assist for migration of nourishment. And one more drawback of this plasmodesma is that viruses can also transmit through this channels. So if one cell gets infected with virus, a plant virus, the same plant virus can cross this tunnel and enter and infect the another cell. So this is about Nux vomica, microscopy and morphology.